Ladies and gentlemen, please welcome Mary Beth Rains to the stage.
Hello, everyone. Okay. So I'm going to get right into it because we don't have that much time. This is actually a slide I repurposed from about 15 years ago when I worked at Quest Diagnostics. And I was fortunate enough to work on some of the first companion diagnostics in the market in my role at Quest. The main difference between a research homebrew assay, even a prototype, even RUO CLIA assay, is there may not be a clinical utility here, but down here there has to be a clinical utility, or we also heard it as a context of use. It may be a multiplex or singleplex. And today I'm going to talk mostly about ultrasensitive singleplexes because that's the most applicable to ligand binding assays. Although Steve and I worked on a multiplex assay, it was 6 independent ELISAs, and it had an algorithm to come up with a score. It was for urine drug-induced acute kidney injury. That was just qualified by the FDA last year. I'm going to talk a little bit about platform selection, and it's very important, obviously, with ultrasensitive assays. Meaningful performance criteria. So what happened at Theranos should never happen at your lab, right? So you want to make sure the CVs are right. And then the regulatory and validation standards, and they're different. They're different in different industries. So the CLSI guy, the CLIA lab validations are a little bit different than the bioanalytical method validations for the FDA versus the EMA. So that fit for purpose becomes really important. And then we heard a little bit this morning earlier about collection and handling and storage. Availability can also be an issue, depending on what your sample type is. Obviously, humerus fluid and some of that is a little bit harder to validate and characterize, and even getting the disease samples can be a challenge, depending on the indication. Assay robustness, I'm going to talk a little bit about assay robustness and lot qualifications. There were some questions earlier, so I have a slide on that. And having that standardization across labs. Ultimately, you're going to start with a single lab up here, but if you have a routine diagnostic test, that's going to be run in multiple laboratories all over the world. And you want the same test result here in Barcelona as you have in Los Angeles. Now, as you go through this development, you may need to make some changes to the assay. And so you need to plan for it. You need to think about it proactively. As you get further down, the less flexible it is. And then, of course, there's multiple stakeholders, and that's where the collaboration and the partners are important. So early on, it may be a fishing expedition, so you may be using a multiplex approach. You may be using commercial kits. As it starts to move into the clinic, it's maybe a single lab that's going to perform that. It should be a defined panel of either one analyte or multiple analytes. There are some pharma companies that like to run, especially in oncology trials, they'll run the same panel across almost every oncology indication that they're looking at. So in that case, there's not as much defined use except that it's going to be run on cancer samples. And then in terms of platform selection, the sample volume, I worked in that for a CRO for many years, turnaround time, and the throughput is very important. And then, of course, selectivity, especially in the oncology and inflammatory diseases. Sensitivity is very important, as well as reproducibility and specificity and selectivity. And then the one thing that's sort of underappreciated is the cost. And the cost, as you get farther to the right, gets a lot more expensive. And I worked for a while at an IVD company, and the cost to develop an IVD, an in vitro diagnostic, is in the millions, tens, is in like $10 million versus a life science company that maybe spent $40K to develop some antibodies and make an ELISA kit. So that's another thing you have to consider as you go through that. And then I talked about the different regulatory guidelines, and really to meet those, you may require modifications, especially if you're using a commercial kit. And then you have to plan for that clinical validation and the context of use as you go through. So a lot of pharma companies, they'll start as an exploratory biomarker, and then they decide they see the first in human, the phase 1 studies, they like the results. Now they want to move it to the next level. And the minute they're going to make a decision off that biomarker is the minute you need to start making sure you have all your regulatory ducks in a row and that they're validated to the right. In some cases, if you're going to actually develop an in vitro diagnostic, you may actually have to change platforms. We heard earlier about taking NFL onto an automated chemistry platform. And that could end up as a in vitro diagnostic. So you have to have a bridging strategy. Even the singleplex that Steve and I worked on, they're working on putting those 6 independent assays as a multiplex assay in one test. And then again, the other thing to consider is once you partner with your IVD company, you're sort of locked into a platform. Once they have their prototype assay, there's no more assay modifications. They have to validate the manufacturing of that kit. And they're interested in the interlaboratory, not within one laboratory, but across many laboratories and that's part of their method validation. So many different partners, stakeholders here. I've actually worked in almost all of these except the large IVD manufacturers in my career. Most of it's been in this middle space here, but having the right partner is very important. And you may end up working with 2 or 3. You may work with pharma, a life science company, or small IVD, as well as the CRO or reference lab. So in terms of when I select, if I'm looking for an ultrasensitive assay, the first thing I do is I look at normal healthy donor data and the percent that's quantifiable. That's above the lower limit of quantitation, which in the Kappa-Clea world is greater. The CVs have to be less than 20%. And so here are here are IL-10 and IL-17 on a couple of different platforms. On the right, you can see the detectability. The quantifiability is the best on the SPX, and it's actually better than on the singleplex Arena platform and better than the ECL platform. IL-10, on the other hand, the levels are lower, and this just goes to show you the difference across methods. So on this ECL platform, calibrators may be different, probably a different antibody pair. Both have decent detectability, but again, the IL-10. You may have matrix effects here, which gives you an inflated value as well. So those are things, but just if I'm just looking, that's the first thing I go look in the catalogs of a commercial assay. Here's a little bit more data on some other ultrasensitives on the SPX platform. Interferon gamma, IL-2, IL-6, IL-17A. You can see there's great separation between the LLOQ and where your normal healthy donor levels are. This is probably one of the most robust ultrasensitive assays on the SPX platform. On the right just shows you the difference you can get in normal ranges. Look, just switching antibodies. So we went from having about 50% detectable, and I said this isn't good enough to the guys at Quanterix, and they came back with another antibody pair, and we went to 100%. So having an open, honest conversation with your vendors can really benefit. This has been my experience with the SPX array, and I also have done a custom assay with them. It was actually one of these inclusion-exclusion. They needed an ultrasensitive platform. And it's in the phase 1 trial. We're determining the cutoff for inclusion-exclusion. This is just some assay performance data with, this is actually in study, so cross studies. You can see the CVs are around 10 to 15%. Usually the low QC is a little bit higher, has a little bit higher CV. We try to drive the sensitivity down. These QCs are, I like to have them, almost all of these are about 10 times higher than the LLOQ, which is a little higher than you would want, but sufficient for the CLIA. Here's some IL-6 ultrasensitive QC performance, and I want to just point out, so these QCs are, there's 2 unspiked serum pools. One is a low, one is a medium level, and the high is spiked. These are used across runs over multi-years. And you can see most of this data is plus or minus 1 SD, which translates into about plus or minus 15% of the nominal value of the QC. This is the benefits of having matrix-based QC. So this is actually an IVD kit, where these two are the kit QCs here. I can see what you can see where the lots changed because you see the shift a little bit in the QCs. These were still within spec. And again, the QC manufacturers will change their limits for their QCs, which is a no-no in CLIA. And here you can see there's a precipitous drop in this QC. And this is our unspiked serum. So we could pick it up a shift with matrix, but we couldn't pick it up on the kit QCs. This is why it's so important to have matrix QCs. This drop ended up being issue with the washer, and that they would manually wash their plates, and we ran it on a plate washer. So I do want to go through this, potential modifications to meet the additional. So we modify the standard curve. I have to speed up because I have a minute left. So we may add more levels to meet the guidelines, so 10 to 12 levels. Should have an anchor point. That's a standard level below the LLOQ. For these ultrasensitive assays, it's very useful to have one at the LLOQ, so you have a very clear cutoff on your standards of where it's below the limit of detection. I like to vary the dilution scheme to cover the AMR and to reduce lab errors. And and then using 21 CFR compliant software like Softmax Pro. The other thing is the reference standard. It's really important to have an external reference standard. As much as the kit standard, you have to keep those commercial kit companies honest, and they like to change the reconstitution volume on the standards between kits, even though it's the same lot. I talked about this. There's all kinds of method changes we make, buffer modifications, MRDs, incubation times, all of these replicates to meet the specs, especially the selectivity, make sure you don't have any matrix effects. And then I'm just going to take this one slide on because my time's up here. So precision, we look at precision and accuracy. I would strongly recommend that you monitor the raw signals and of both the QC and the standards. Don't just look at the back calculated because the signals are important. That's the way you can troubleshoot your assay. For the highly sensitive assays, you really want to make sure your background, do what you can to keep the background levels from changing. If your QCs fail, you think they're degraded, go back and look at your normal healthy donors because the reference range shouldn't change over time. It should always be the same. In the IVD world, we use commutability samples to qualify lots. And it can be as simple as the EMA and the FDA bioanalytical guidelines to just use 5 levels of the one at the upper limit of quantitation, one at the lower, or you can run 20 different matrix samples across the curve, across the standard curve, and then use regression analysis. And then finally, this having a good strategic partnership and really proactive communication and making sure they let you know of any method changes that they anticipate and really giving them constructive, and the key word there is constructive. And, figure out if you need to do any customizations, share data and samples. That's what we did with the IVD company to troubleshoot that. And then my big thing now is really to see if you could work up front to purchase and sequester larger antibody lots to minimize changes in antibody, because that's the biggest problem with lot-to-lot variation is if they change antibodies. And so, and with that, I think I'll stop. I have one more slide, but it's just a summary.